Okay, let's unpack this. Today, we're embarking on a really fascinating deep dive into the very building blocks of plant life.
That's right.
We've got a stack of incredible insights here from Raven Biology of Plants, specifically Section 1. It lays out this incredible world of the plant cell, from the tiniest molecules right up to the grand ballet of cell division.
It really is quite a dance.
So our mission for you, our listener, is to give you a shortcut, sort of to being truly well-informed about the fundamental chemistry and structures that make plants tick, all without needing to, squint at a textbook diagram.
Exactly. Think of it as peeling back the layers.
Yeah, seeing the vibrant, dynamic universe inside every leaf and root, and understanding why it all matters.
Indeed. We'll explore the remarkable molecular composition first. That's the blueprint for all living things, really. Then we'll zoom in on the intricate machinery of the plant cell itself. And finally, we'll consider how substances actually move in and out of these vital units.
Right, because it's not just static.
Not at all. And this isn't just about, memorizing parts. It's about understanding the why behind the what and how these foundational concepts connect to the bigger picture of how plants sustain life on Earth.
Okay.
You'll gain, hopefully, a thorough understanding of these complex processes presented in a way that really brings them to life. Maybe even get a few of those aha moments that make learning stick.
Love those moments. All right, let's jump in. #tag the molecular composition of plant cells. So if we're going to understand the molecular drama of a plant cell, where do we even begin? I'm guessing it all starts with the most basic ingredients, the elements themselves.
Yeah, right. It absolutely does. I mean, while our planet boasts 92 naturally occurring elements, evolution, in its incredible efficiency, it selected just 6.
Only 6.
Yeah, just 6: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur.
Yeah.
You often hear that acronym, CHNOPS, right?
CHNOPS, got it.
These 6 make up a staggering 99% of the weight of all living matter. They're the real stars of our molecular cast.
Wow, 99% from just 6. That's amazing.
It really is. And here's another surprising fact. Over half of all living matter, and actually more than 90% of most plant tissues, is simply water. H2O.
Just water.
It's a powerful reminder of how fundamental that simple molecule is to life itself. Almost everything else you find in a living organism, chemically speaking, is composed of organic molecules, those fascinating carbon-containing compounds that truly form the building blocks.
It's incredible to think that all the complexity of a plant comes from just these few elements, often bound together in these carbon-based compounds. So these thousands of different organic molecules in a plant cell, they can't just be random, right? They must fall into some major category.
Exactly right. Despite the vast number and variety, they can be grouped into 4 main types that play the major roles in living systems. We're talking carbohydrates, lipids, proteins, and nucleic acids.
The big 4.
The big 4. And what's truly fascinating here is how these large molecules, which we call macromolecules, things like polysaccharides, proteins, and nucleic acids, are typically built. They're constructed from smaller, similar subunits called monomers.
Okay, like building blocks.
Think of them exactly like individual Lego blocks. The cell links these blocks together through a process called dehydration synthesis. Basically, a molecule of water is removed for each new bond formed.
Water out, link forms.
Precisely. And the reverse, breaking these large molecules down into their individual subunits, that's called hydrolysis. Hydro, water, hydrolysis, breaking. So a water molecule is added to break a bond.
Water in, link breaks. Simple but powerful.
It's a fundamental principle.
Yeah.
The simple add-remove water mechanism. It's how cells build and break down their essential components, constantly recycling resources.
Okay, let's dive into carbohydrates then. We often think of them as just, sugar for energy, but they do so much more, don't they? What's their main role in the plant world?
Oh, far from just sugar rushes. Carbohydrates are, in fact, the most abundant organic molecules in nature, so hugely
Or
important.
most abundant, wow.
Yeah, they serve 2 absolutely critical roles in plants and, well, all living organisms. First, as primary energy storage. Second, as crucial structural components.
Energy and structure.
Exactly. For instance, the walls of young plant cells, the things that give them their shape and strength, built largely from cellulose, a universally important carbohydrate. It's embedded in a matrix of other stuff, but cellulose is key. So yeah, carbs are about both fuel and fundamental architecture.
And these come in different forms, like from tiny single units to giant complex chains.
Yes, you can categorize them by their sugar subunits. Makes sense, right?
Uh-huh.
So first we have monosaccharides, or single sugars. They're glucose, fructose. They're the fundamental building blocks, simple molecules, easily dissolved in water, and they're a primary immediate source of chemical energy for plants and animals alike.
The quick energy hit.
Kinda, yeah.
Yeah.
Then you have disaccharides, or two sugars linked together. Sucrose, common table sugar, that's a prime example, made of glucose and fructose linked up. It's the form in which sugar is efficiently transported in most plants.
Transported.
Yeah, moving from the photosynthetic leaves, where it's made, to other parts of the plant body that need energy, like growing roots or maybe developing fruits.
Ah, okay. Makes sense.
And finally, we have polysaccharides. Poly meaning many. So many sugars, long chains of linked monosaccharides. These serve really diverse functions.
Okay, like what?
Well, for storage, the main energy reserve in plants is starch. It's made of many alpha-glucose molecules. You can sort of visualize starch in two ways. There's amylose, which is like a uniform coil, kind of like a spring.
Okay.
And then there's amylopectin, which is more branched, like a little tree structure.
Spring and tree. Got it.
Starch is stored as visible grains within plant cells, ready to be broken down by hydrolysis back into usable sugars when energy is needed. Oh, and there's a lesser-known storage form, too, fructans. They're polymers of fructose found in grains like wheat. They're water-soluble and can actually be stored in much higher concentrations than starch, which is an advantage sometimes.
Interesting. And the structural ones, you mentioned cellulose.
Right, the structural polysaccharides. This is where plants truly excel at building. Cellulose is the star here. It's the principal component of the plant cell wall.
The wall again?
Yep. It's also a polymer of glucose, like starch, but it uses a different form, beta-glucose. And this seemingly small difference makes its chains long, unbranched, and incredibly straight.
Straight chains.
And these straight chains bundle together to form these incredibly strong microfibrils. They get woven into the cell wall, making it incredibly rigid and strong. We're talking tensile strength that can actually exceed steel, pound for pound.
Stronger than steel. That's wild.
It is. Cellulose is the most abundant organic compound on Earth, making up about half of wood. And unlike starch, most organisms actually can't break down cellulose easily. They need specialized microorganisms to do it.
Right. So besides cellulose.
Well, beyond cellulose, there are other structural polysaccharides like hemicelluloses and pectins. They form a crucial matrix around the cellulose in the cell walls. Pectins, you might know them, they're plentiful in fruits, and they're what give jams and jellies their thickening properties.
Ah, so that's pectin.
That's pectin. And they also act as the cement that holds adjacent plant cells together. That layer is called the middle lamella.
Oh, and there's even chitin, a modified sugar molecule with nitrogen that forms the cell walls of fungi and, the exoskeletons of insects.
Wow.
It's a testament, really, to how slight chemical tweaks in these sugar units can lead to dramatically different functions, from quick energy to super rigid architecture.
That's a great illustration of how a simple building block can be put to such varied uses. Okay, moving from carbohydrates, we have lipids, usually fats and oils. They're called water-fearing hydrophobic, meaning they repel water. What makes this water-fearing quality so special and essential for a plant?
Yeah, their hydrophobicity is absolutely key to their functions. Lipids are primarily for two main things: highly efficient energy storage, like the oils you find packed into seeds.
Right, like sunflower seeds. Exactly. And also for crucial structural purposes, especially in membranes and protective layers. Unlike carbs and proteins, they aren't strictly polymers of repeating monomers in the same way, but they are still large, complex molecules.
So fats and oils are for storing energy, but what actually differentiates a fat from an oil? Is it just solid versus liquid?
That's a big part of it. Fats and oils are both triglycerides. That means they're made of 3 fatty acid molecules bonded to one glycerol molecule. They're nonpolar, and that's why they're hydrophobic. The difference you asked about fat versus oil often comes down to their physical state at room temperature.
Okay.
And that state is determined by their fatty acid chains. Saturated fats have no double bonds between the carbon atoms in their fatty acid chains. This makes the chains straight so they can pack closely together.
Like stacking logs.
Kind of, yeah. So they tend to be solid at room temperature, like animal fats, butter. Unsaturated fats, on the other hand, contain one or more double bonds. These double bonds create kinks, or bends, in the hydrocarbon chain.
Kinks. So they don't stack well.
Exactly. These kinks prevent close packing, which results in a lower melting point. So unsaturated fats are typically liquid. We call them oils at room temperature. Think olive oil, safflower oil from plants.
Ah, the kinks make the difference.
They do. And what's more, fats and oils pack a lot of energy, significantly more per gram than carbohydrates or proteins. That's because they have a higher proportion of those energy-rich carbon-hydrogen bonds.
Makes sense for storage then. And what about their role in membranes? I often hear the term phospholipid bilayer. What's that all about?
Ah, yes, the phospholipids. They are perhaps the most vital structural lipids. They're basically modified triglycerides, where one fatty acid is replaced by a phosphate group. And that phosphate group is negatively charged, making that end of the molecule hydrophilic or water-loving.
So one end loves water, the other hates it.
Precisely. You have a molecule with a hydrophilic head that loves water and two hydrophobic tails, the fatty acid chains, that fear it. Now, when you put these molecules in water, they spontaneously arrange themselves into a phospholipid bilayer.
How does that work?
Well, the hydrophilic heads face outward towards the water on both sides of the layer, and the hydrophobic tails tuck inward, away from the water, facing each other in the middle.
Okay, like a sandwich.
Kind of. Or imagine a double layer of tiny swimmers all facing out to the water with their heads, keeping their legs dry in the middle. This self-assembling bilayer forms the fundamental structural basis of all cellular membranes. It essentially controls what goes in and out of the cell. It's incredibly important.
Wow, self-assembling. That's cool. Beyond membranes, lipids also create protective barriers, which sounds crucial for a plant just sitting out there in the environment.
Absolutely. Cutin, suberin, and waxes are unique lipids crucial for forming barrier layers on plant surfaces, mainly helping to prevent water loss.
How so?
Well, for instance, a protective layer called the cuticle, which is made of wax embedded in cutin, covers the outer walls of leaves and stems. You know that shiny layer you can sometimes polish off an apple? That's epicuticular wax. Part of the cuticle. Suberin is another one. It forms layers in cork cells like the ones in tree bark, also helping to prevent water loss and providing protection. And waxes themselves are just generally the most water repellent of the lipids, providing essential protection for plants against drying out.
Okay, vital waterproofing. And then there are steroids, known for their distinctive four-ring structure. Do plants use these in the same way animals use, say, cholesterol?
Steroids are indeed distinctive, characterized by those four interconnected hydrocarbon rings. While cholesterol is common in animals, plants primarily contain related steroids called sterols, like sitosterol.
Sitosterol.
Right. These are important components of cellular membranes, where they actually insert themselves among those phospholipid tails we just talked about. They help stabilize the tails and influence membrane fluidity, kind of regulating how stiff or fluid the membrane is.
Ah, membrane regulators.
Exactly. And some steroids can even act as hormones in plants, influencing growth, or in some lower plant forms, even serving as sex attractants. So these multi-ring lipids play crucial structural and signaling roles across various organisms.
Okay, fascinating. Let's move on to proteins. They seem to do everything in the cell. What's their core identity given such diverse roles? How can one type of molecule do so much?
You're absolutely right. Proteins are incredibly versatile. They're among the most abundant organic molecules, making up 50% or more of the dry weight of most organisms. They perform an astonishing diversity of functions. They can be structural components, enzymes that catalyze reactions, transporters, storage molecules, signaling molecules. The list goes on.
So what's the common thread?
The common thread is their structure. In their basic structure, proteins all follow the same blueprint. They are all polymers of nitrogen-containing molecules called amino acids, arranged in a specific linear sequence.
Amino acids. Okay, and how many different amino acids are we talking about here?
A lot. Surprisingly, no. Just 20. It's quite remarkable. Whether you're looking at a bacterium, a human, or an oak tree, it's always the same. 20 different kinds of amino acids that are used to form all the thousands of different proteins an organism might synthesize.
Only 20. How do they differ?
Each amino acid has a central carbon atom bonded to an amino group, a carboxyl group, a hydrogen atom, and then a unique R group, or side chain. This R group is the key difference. It determines the amino acid's specific properties, like whether it's polar or nonpolar, charged or uncharged. And that, in turn, largely dictates how the final protein will fold up and interact with other molecules.
And how do these amino acids link up to form a protein?
They link up through peptide bonds. This is another example of that dehydration synthesis we talked about earlier, water out, link forms. The amino group of one amino acid links to the carboxyl group of another, forming a long chain called a polypeptide. Proteins are essentially one or more large polypeptides, often consisting of hundreds or even thousands of amino acids.
So the sequence is key.
Absolutely. The sheer number of possible sequences you can make with just 20 different amino acid letters allows for immense variety. It's much like how our 26-letter alphabet can form countless different words and sentences.
So the sequence matters hugely, but what about the shape? I always hear that a protein's shape is critical for its function, and something about levels of organization.
This is absolutely crucial, yes. A protein's function is intimately, inextricably tied to its precise three-dimensional shape. If you mess up the shape, you usually mess up the function. We describe this intricate organization in 4 hierarchical levels.
Okay, level 1.
The primary structure. This is simply the linear sequence of amino acids in the polypeptide chain. Think of it like a specific word written with amino acid letters. This sequence is dictated directly by the cell's genetic information, and even a tiny change, like substituting one amino acid for another, can sometimes dramatically alter or even destroy the protein's function.
Right. Sequence first, then.
Then, as the polypeptide chain is being assembled, local interactions, primarily hydrogen bonds between parts of the peptide backbone, cause it to fold into recurring patterns. This is the secondary structure. The most common patterns are the alpha helix, which is a coiled shape like a spiral staircase.
Yes, a coil.
And the beta-pleated sheet, where segments of the chain lie parallel to each other in a sort of zigzag sheet-like arrangement. Proteins that exist mostly in these forms, like keratin in hair or silk, are called fibrous proteins. They tend to provide structural support and shape.
Helix and sheet. Got it. Next level.
Tertiary structure. For most proteins, especially those with active roles like enzymes, the secondary structures fold further into a complex, compact, specific three-dimensional shape. This folding is driven by all sorts of interactions among the diverse R groups, attractions between positive and negative charges, hydrophobic interactions pushing non-polar groups together away from water, hydrogen bonds, and even strong covalent bonds called disulfide bridges that can form between sulfur-containing amino acids, locking parts of the molecule firmly in place. Most biologically active proteins, like enzymes, are globular proteins with complex tertiary structures.
So that's the final 3D shape.
For a single polypeptide, yes. And here's where it gets really interesting. If this intricate tertiary shape is disrupted, say by heat or extreme pH changes, the protein unfolds. This is called denaturation.
Ah, like cooking an egg white.
Exactly like cooking an egg white.
Uh-huh.
The protein unfolds, loses its specific 3D structure, and consequently it loses its biological activity. It usually can't go back.
Okay. And the 4th level.
The quaternary structure. This applies only to proteins that are made up of more than one polypeptide chain. The quaternary structure describes how these individual polypeptide chains, or subunits, interact and associate with each other to form a single, larger, functional protein molecule. Think of hemoglobin, which carries oxygen in our blood. It's made of 4 polypeptide subunits working together. These interactions can involve hydrogen bonds, hydrophobic forces, or disulfide bridges, holding the whole complex together.
Okay, 4 levels to get the final working protein. And speaking of functions, enzymes are super important, aren't they? You called them catalysts. What does that really mean?
Absolutely vital. Enzymes are typically large, complex, globular proteins that act as incredibly efficient biological catalysts. By definition, a catalyst is a substance that speeds up the rate of a chemical reaction without being consumed or changed itself in the process.
How do they speed things up?
They do it by lowering the energy of activation required for the reaction to occur. Think of it like needing less of a push to get a boulder rolling downhill. Enzymes provide an alternative reaction pathway that requires less initial energy input.
So they make reactions happen much more easily.
Exactly. This means they can be used over and over again, making them incredibly efficient even at very low concentrations within the cell. Each enzyme is typically highly specific, usually catalyzing only one or a very small number of related chemical reactions. Without enzymes, the chemical reactions necessary for life would simply occur far too slowly to sustain a living cell, especially at the moderate temperatures found in most organisms. They often get their names by adding "ase" to the name of the substance they act upon, like amylase breaking down amylose, which is starch.
Right, amylase for amylose. Okay. So if proteins are the versatile workers and enzymes are the catalysts making things happen, then nucleic acids. They sound like the architects carrying all the instructions, right? What are their fundamental building blocks?
That's a fantastic analogy, yes. Nucleic acids are the molecules that encode and then help translate the genetic information. That information dictates the structures of all those thousands of different proteins we just discussed and ultimately controls cell structure and function. Like proteins, nucleic acids are polymers, long chains, but their building blocks are different, slightly more complex molecules called nucleotides.
Nucleotides. Okay, and what exactly is a nucleotide made of?
A nucleotide itself has three distinct components chemically bonded together. There's a phosphate group, there's a 5-carbon sugar, which could be either ribose or deoxyribose, and that difference is important. And finally, there's a nitrogen-containing base, often just called a nitrogenous base.
Phosphate, sugar, base.
Right. There are 5 main types of these nitrogenous bases: adenine A, guanine G, thymine T, cytosine C, and uracil U. These nucleotides then link together again through dehydration synthesis, removing water to form the bonds, creating long chains or polynucleotides.
So DNA and RNA are these long chains of nucleotides, but with key differences based on the sugar and bases.
Exactly. Ribonucleic acid, or RNA, has ribose as its 5-carbon sugar subunit. It also uses the base uracil, U, instead of thymine, T. And RNA typically consists of a single chain of nucleotides, although it can fold back on itself sometimes.
Okay, ribose, uracil, single chain for RNA and DNA.
Deoxyribonucleic acid, or DNA, uses deoxyribose as its sugar that's ribose with one less oxygen atom, hence deoxy. It uses thymine T instead of uracil. And DNA molecules are famously long, typically consisting of 2 chains coiled around each other in that iconic double helix structure. DNA molecules are generally the largest macromolecules found in cells.
And their roles relate to this structure.
Absolutely. DNA is the primary carrier of the genetic messages. It contains the hereditary information organized into units called genes that organisms inherit from their parents. RNA molecules are primarily involved in the process of translating this genetic information from the DNA blueprint into the actual synthesis of proteins. Though, interestingly, some RNA molecules can even fold up and function as ribozymes, which are RNA molecules with enzyme-like catalytic activity.
RNA acting like an enzyme. Cool. And where does ATP fit into all this? I know it's often called the energy currency of the cell. Is it related to nucleic acids?
That's a very perceptive question, and it beautifully connects molecules to energy flow. Yes, ATP, or adenosine triphosphate, is actually a modified nucleotide. Think of it as an adenosine monophosphate, AMP, which is a nucleotide with adenine, ribose, and 1 phosphate, but with 2 additional phosphate groups tacked on.
So 3 phosphates in total.
Exactly, triphosphate. The bonds linking these last 2 phosphate groups are relatively weak, sometimes called high-energy bonds, though that's a bit misleading. When the terminal phosphate bond is broken by hydrolysis, adding water, it releases a significant amount of free energy. This converts ATP to ADP, adenosine diphosphate, 2 phosphates, and a free inorganic phosphate molecule.
And the cell uses that released energy.
Precisely. That readily available packet of energy released from ATP hydrolysis is used to power countless energy-requiring chemical reactions within the cell. Everything from building complex molecules to transporting substances across membranes to muscle contraction or in plants, things like cytoplasmic streaming, is truly like the cell's rechargeable battery. ADP gets recharged back to ATP using energy capture during energy-releasing processes like cellular respiration or photosynthesis. So ATP is the molecule directly involved in providing the immediate energy for most of the living cell's activities.
Okay, that makes sense. Now, this is where plants get really clever, I think. Beyond the basic molecules we all need to live, carbs, lipids, proteins, nucleic acids, plants produce all sorts of other amazing compounds, don't they? What's the big picture insight into these secondary compounds?
They certainly do, and this is where plants truly reveal their evolutionary ingenuity and complex chemistry. Historically, compounds essential for the basic life processes of all plant cells, like simple sugars, amino acids, proteins, were called primary metabolites.
Essential.
Right, but then there's secondary metabolites. These are compounds that are more restricted in their distribution. You don't find them in all plant cells, or even in all plant species. They might be specific to certain families or genera, or even specific tissues within a plant.
And they aren't just waste products.
Not at all. That was an old idea. We now know they are absolutely crucial for a plant's survival and propagation, often in surprisingly sophisticated ways. The core insight here is that plants have evolved this incredibly sophisticated chemical arsenal beyond basic survival needs. They use these compounds for defense, communication, attracting pollinators, deterring herbivores, and even adapting specifically to their local environment. They're like the plant's specialized tools or weapons.
So these secondary compounds aren't just for show then. They have vital, often surprising roles.
Absolutely. Many act as chemical signals, enabling plants to respond to environmental cues or communicate with other organisms, like beneficial microbes in the soil. Others are critical for defense against herbivores, making leaves taste bitter or even poisonous, or defense against pathogens like fungi and bacteria. Some even inhibit the growth of competing plants nearby, a process called allelopathy.
Chemical warfare.
In a way, yes. Some secondary metabolites provide protection from harmful UV radiation from the sun, acting like sunscreen, and still others aid in attracting animals for pollen and seed dispersal, using scents or colors derived from these compounds. They're often produced in specific plant parts and sometimes stored safely in vacuoles to prevent the plant from poisoning itself. There are three major classes we usually talk about: alkaloids, terpenoids, and phenolics.
Okay, let's start with alkaloids. I know some famous ones there, often with powerful effects on humans.
You're right. Alkaloids are perhaps best known for their often dramatic physiological and sometimes psychological effects on humans, which makes them pharmacologically and medicinally significant, but also potentially dangerous.
It's like, right. Well, think of morphine, isolated from the opium poppy. It's a powerful analgesic, a painkiller, and also a cough suppressant, though highly addictive. Or caffeine, a stimulant found in coffee, tea, and cocoa. Interestingly, in the coffee plant itself, high concentrations of caffeine in seedlings are actually toxic to insects and fungi.
Defense.
Exactly. And it can even inhibit the germination of other seeds nearby. That's the allelopathy we mentioned, helping the coffee seedling avoid competition. Then there's nicotine from tobacco leaves. It's a highly toxic stimulant that strongly deters most herbivores and insects. And get this, nicotine synthesis increases in response to wounding, acting as a phytoalexin, which is an antimicrobial compound produced by the plant to fight off infection after damage.
Wow, they ramp up production when attacked.
They do. And there are many others, like atropine, famously used by Cleopatra to dilate her pupils, but now used medically as a cardiac stimulant and even an antidote for nerve gas. These examples truly highlight the potent chemical arsenal plants possess, often evolved primarily for their own defense or ecological interactions. Okay, powerful stuff. Next up, terpenoids. You said this was the largest class. What are they all about?
Terpenoids, or sometimes called terpenes, are by far the largest class of secondary metabolites, with over 22,000 described compounds. And they're all built from simple, repeating 5 carbon units derived from isoprene, C5H8.
Isoprene.
Isoprene itself is actually a gas. It's emitted by many plants, especially trees like oaks and poplars, particularly on hot days. It's largely responsible for that bluish haze you sometimes see over wooded hills in the summer.
The haze is isoprene.
Mostly, yeah. And plants seem to use this isoprene blanket to help them cope with heat stress, possibly by stabilizing photosynthetic membranes in the leaves.
Interesting. What about other terpenoids?
Well, many smaller terpenoids, like monoterpenoids, 2 isoprene units, and sesquiterpenoids, 3 units, are volatile and known as essential oils. They contribute to the characteristic fragrance of plants. Think of menthol and mint, limon and citrus, pine and pine trees. These oils can deter herbivores, protect against fungi, or conversely attract pollinators or seed dispersers.
So scents and flavor.
Often, yes. Then there are larger ones. A terpenoid, 4 isoprene units, called Taxol, originally found in the bark of the Pacific yew tree, has gained significant attention for its potent anti-cancer properties.
Taxol, right, the cancer drug.
That's the one. Sourcing it originally was problematic because it killed the tree, but scientists have since found it in other sources, like the needles of other yew species, and even in a fungus that lives on yews, and importantly, they've figured out how to synthesize it in the lab. It's a great example of how we rely on plant chemistry for critical medicines.
Absolutely.
And here's an amazing ecological aha moment involving terpenoids. Monarch butterfly caterpillars feed almost exclusively on milkweed plants. Milkweeds contain highly poisonous terpenoids called cardiac glycosides. These are sterile derivatives that can cause heart attacks in vertebrates.
Poisonous, okay.
But the monarch caterpillars are adapted. They ingest and store these toxins in their bodies without being harmed. This makes both the caterpillar and the adult butterfly poisonous to predators, mainly birds.
So the butterfly becomes poisonous by eating the plant.
Exactly. And the monarch's bright orange and black coloration then serves as a conspicuous warning signal: don't eat me, I taste bad and might kill you.
Eee.
Birds quickly learn to avoid them after just one unpleasant experience. It's a classic example of chemical defense and warning coloration. Oh, and the largest known terpenoid is rubber, a huge polymer of hundreds or thousands of isoprene units harvested from the latex of the tropical rubber tree.
Rubber is a terpenoid. Wow. Okay, and finally phenolics. What kind of compounds are in this broad group?
Phenolics are another very broad and important class. They're all characterized chemically by having a hydroxyl AH group attached directly to an aromatic ring, a benzene-like ring structure. They're found universally in plants and accumulate in pretty much all plant parts.
What do they do?
Lots of things. One major group within phenolics are the flavonoids. These are often water-soluble pigments frequently found stored in the vacuoles. They include the anthocyanins.
Anthocyanins, those sound familiar.
They probably do. Anthocyanins are responsible for many of the beautiful reds, purples, pinks, and blues you see in flowers, fruits, like grapes, berries, red cabbage, and even the brilliant red colors of some autumn leaves. Other flavonoids, like flavones and flavanols, are typically yellowish or even colorless, but they can sometimes complex with anthocyanins and metal ions in a process called copigmentation to create intensely blue flower colors, which are otherwise quite rare.
So they're pigments attracting pollinators.
Yes, attracting pollinators and seed dispersers is a key role. Flavonoids also interact with beneficial soil bacteria, help plants defend against microbes, and provide important protection against damage from UV radiation.
Multitaskers. What else in phenolics?
Then there are tannins. These are phenolic compounds that act as significant deterrents to herbivores because they have a very astringent, bitter taste. They're often found in high concentrations in unripe fruits, think of that puckering taste, or in the bark and leaves of trees like oaks. Humans, of course, have historically used tannins extracted from plants to tan animal hides into leather. The tannins bind to the proteins in the hide, making it more resistant to decay.
Ah, tannins for tanning leather. Makes sense.
Exactly. And then there's a hugely important group of phenolics called lignins. Lignins are perhaps the second most abundant group of organic compounds on Earth, right after cellulose. Unlike most other secondary metabolites, lignins aren't stored in the vacuole. They are deposited within the cell wall itself, particularly in secondary walls.
What do lignins do in the wall?
They contribute immense compressive strength, stiffness, and also waterproofing to plant tissues, especially wood. The process of lignification, adding lignin to cell walls, was absolutely crucial for the evolution of large, upright terrestrial plants. It allowed them to grow tall, supporting large photosynthetic surfaces, and overcoming the forces of gravity. Lignin also helps make the walls impermeable to water, which is essential for efficient water transport in xylem tissue, and it acts as a physical barrier against pathogen attack. Sometimes wound lignin is formed after injury to seal off damaged areas.
So lignin is key for being a tree, basically.
Pretty much, yeah. And one last phenolic to mention, salicylic acid. This might ring a bell as the active chemical relative of aspirin.
Aspirin, right, from willow bark.
Originally, yes, derived from compounds found in willow bark used for centuries for pain relief. But in the plant itself, salicylic acid plays a crucial role as an internal signaling molecule. It's essential for triggering systemic acquired resistance.
Systemic acquired resistance.
Yeah, it's like a plant-wide immune response. After a localized attack by a pathogen, say on one leaf, salicylic acid signaling helps induce a long-lasting state of heightened defense readiness throughout the entire plant, making other parts more resistant to subsequent infections. It's the plant's own internal alarm system and defense booster. These secondary metabolites really showcase the remarkable, often hidden chemistry driving plant life, defense, and interactions. #tag the plant cell in the cell cycle.
Okay, so from all those molecules, we finally jump up a level to cells, the actual fundamental units of life. Robert Hooke famously coined the term, didn't he, looking at cork? And what's the big idea behind cells being the fundamental unit? Why is that theory so important?
That's right, back in the 17th century, Robert Hooke peered through his early microscope at a thin slice of cork and saw all these tiny empty compartments that reminded him of the small rooms, or cells, in a monastery. So he called them cells.
But they were empty, just the walls.
In cork, yes, because cork is dead tissue. It took another, 150 years or so, with better microscopes and more observations, for the term to take on its modern biological meaning as the basic structural and functional unit of living matter. The cell theory, developed mainly by Schleiden, Schwann, and Virchow in the 19th century, became a unifying theme for all of biology.
And what does it state, basically?
It essentially states three main things. First, all living organisms are composed of one or more cells. Second, all the chemical reactions of life occur within cells. And third, all cells arise only from pre-existing cells through division. Later, the idea that cells contain the hereditary information, DNA, passed from generation to generation, was added. It really established the cell as the ultimate self-contained unit for life.
Okay, the cell as the basic unit. But I've heard that for plants, there was also this slightly different idea, an organismal theory. Why was that considered important, especially for plants?
Indeed. While the cell theory emphasizes the individual cell as the fundamental unit, the organismal theory offers a complementary perspective. particularly relevant to plants and some other multicellular organisms. It suggests that the entire organism, not just the individual cells composing it, is the basic unit of life.
How does that work for plants?
Well, it sees a plant not just as a collection of independent cells, but rather as a continuous mass of living contents, the protoplasm, which is then subdivided into cellular compartments. This view is particularly applicable to plants because, as we'll see, their cells don't completely pinch apart during division like animal cells often do. Instead, their living contents, the protoplasts, that's everything inside the cell wall, remain physically interconnected.
Interconnected how?
They're connected by thousands of tiny cytoplasmic strands called plasmodesmata that pass through the cell walls. These plasmodesmata effectively link the cytoplasm of nearly all the cells throughout the plant body into one continuous network. This interconnected network is called the symplast.
Symplast, like one giant interconnected living system.
Exactly. So while plants are clearly made of distinct cells, these cells are so deeply interconnected that the organismal theory, emphasizing the unity of the whole plant, provides a very valid and important perspective. The plant truly functions as a coordinated whole.
Okay, so we have cells. They're fundamental but also interconnected in plants. Now, thinking about cell types broadly, there are two fundamental kinds of cells out there in the living world, right? What's the big defining difference between them?
Yes, there's a really fundamental split in how life is organized at the cellular level. The main distinction, the really big one, is the presence or absence of a true membrane-bound nucleus.
Nucleus or no nucleus?
That's the key divide. On one side, you have the prokaryotes. The name means before and nucleus. Pro meaning before, karyon meaning kernel or nucleus. This group includes bacteria and archaea. Their genetic material, typically a large circular DNA molecule, is localized in a region of the cytoplasm called the nucleoid, but crucially, it's not enclosed by a membrane envelope.
Okay, DNA just in the cytoplasm, basically.
Pretty much, yeah, in that nucleoid region. Yes. Prokaryotic cells also notably lack most other types of specialized membrane-bound internal compartments or organelles that you find in the other group. They are generally simpler in structure.
And the other group?
That's the eukaryotes. The name means with a true nucleus, meaning true. This group includes all plants, animals, fungi, and protists. Their defining feature is that their genetic material, the DNA, is organized into multiple linear chromosomes, and these chromosomes are contained within a well-defined nucleus that is enclosed by a double membrane, the nuclear envelope.
Ah, the DNA is packaged away safely.
Exactly. And eukaryotic cells also boast numerous other specialized membrane-bound organelles suspended in their cytoplasm, things like mitochondria, chloroplasts in plants and algae, the endoplasmic reticulum, the Golgi apparatus, and so on. Each organelle performs distinct functions. This internal compartmentation by membranes is a key hallmark of eukaryotes, allowing for much greater structural complexity and functional specialization within the cell. It's a more complex and organized way to run the business of life.
Okay, so plants are eukaryotes with a true nucleus and all those organelles. So what does a typical plant cell look like on the inside then, beyond just having a nucleus? What are its key components?
All right, let's picture a typical plant cell. Starting from the outside, you usually have that relatively rigid cell wall.
The defining feature.
Absolutely. Inside the cell wall, you have the living part, the protoplast. The protoplast consists of the cytoplasm and embedded within the cytoplasm, the nucleus.
Okay, wall, then protoplast, which is cytoplasm plus nucleus.
Right. The cytoplasm itself is where most of the action happens. It's not just empty soup, it's a bustling factory. It contains all those specialized membrane-bound organelles we just mentioned, like plastids, including chloroplasts, and mitochondria. It also has intricate membrane systems like the endoplasmic reticulum, ER, and the Golgi apparatus. And then there are non-membranous structures too, like ribosomes for protein synthesis, and elements of the cytoskeleton like actin filaments and microtubules, which provide structure and allow movement.
And the soupy part.
Ah yes, the watery cellular soup in which all these organelles and structures are suspended is called the cytosol. And the entire cytoplasm, soup, and all the bits within it is enclosed by the plasma membrane. This plasma membrane lies just inside the cell wall and acts as that crucial, selectively permeable gatekeeper, controlling what enters and leaves the living protoplast.
Okay, quite a complex interior. And you mentioned earlier that plant cells have some distinct features that really set them apart from, say, animal cells. What were those big three again?
Yes, absolutely. While they share many features with other eukaryotic cells, plant cells typically have three key structures that fundamentally distinguish them from most animal cells. First, as we keep saying, there's that rigid cell wall on the outside providing support and protection.
Check.
Second, they possess plastids. This is a whole family of organelles unique to plants and algae. The most famous type, of course, is the chloroplast, the site of photosynthesis, but there are other types too, involved in storage or pigment synthesis.
Plastids, got it. And the third?
The third is the presence of one or more, often very large, liquid-filled vacuoles. In a mature plant cell, a single central vacuole can take up a huge portion of the cell volume, like 80 or even 90%. These vacuoles are enclosed by their own single membrane, which has a specific name, the tonoplast.
Wow, 90% just vacuole. Okay, so wall, plastids, and large vacuoles. Those are the plant cells that you mentioned.
Those are the big three, yes. And another thing you often see specifically in living plant cells, if you look under a microscope, is that the contents of the cytoplasm, including organelles like chloroplasts, aren't just sitting still. They're often actively flowing or sweeping along in an orderly, sometimes quite rapid, continuous fashion around the edge of the cell.
Things are moving inside.
Oh yeah. This fascinating movement is known as cytoplasmic streaming, or sometimes cyclosis. It's driven by the cytoskeleton, and it helps to circulate materials within the relatively large volume of the plant cell, ensuring efficient transport and distribution of nutrients and metabolites. It's quite dynamic.
Cool. Okay, let's zoom in on some of those components. The nucleus, you said, it's often the most prominent part. It's basically the cell's brain or control center, right? What are its primary functions?
It is very much the control center, yes.
Yeah.
You can think of it as having two absolutely vital interconnected functions. First, it controls the cell's ongoing activities, its metabolism, its growth, its responses to stimuli. It does this mainly by dictating which specific protein molecules are produced by the cell, in what amounts and when. Proteins, especially enzymes, carry out most of the cell's work, so controlling protein synthesis is key to controlling the cell.
Okay. It controls protein production. And the second function?
The second function is to serve as the cell's primary genetic vault. It stores the vast majority of the cell's hereditary information, its nuclear genome, encoded in the DNA molecules that make up the chromosomes. And crucially, it must be able to accurately replicate this information and faithfully pass it on to daughter cells during cell division, ensuring continuity from one generation to the next.
So day-to-day control and long-term inheritance.
Exactly. Although it's worth remembering, as we mentioned, that in plant cells, the nucleus doesn't hold all the genetic information. The plastids, like chloroplasts, and the mitochondria also contain their own smaller, independent sets of DNA and genes, reflecting their unique evolutionary origins. But the nucleus is definitely the main library.
Right, the semi-autonomous organelles. And the nucleus itself is surrounded by its own specialized membrane.
Yes, it is enclosed by a distinctive nuclear envelope, which is actually a double membrane, 2 phospholipid bilayers thick. So there's an inner nuclear membrane and an outer nuclear membrane, separated by a narrow space.
A double barrier. Is it sealed off?
Not completely. This envelope isn't a solid, impermeable barrier. It's actually perforated by numerous intricate structures called nuclear pores. These aren't just simple holes, they are complex protein structures that act as highly regulated passageways. They control the crucial traffic of molecules like messenger RNA going out to the cytoplasm and proteins needed inside the nucleus coming in between the nucleus and the surrounding cytoplasm.
Gateways in the double wall.
Precisely. And interestingly, the outer membrane of the nuclear envelope can often be seen to be continuous with another membrane system in the cytoplasm, the endoplasmic reticulum, which we'll get to. Inside the nucleus, the DNA isn't just floating around loose. It's organized together with various proteins, primarily basic proteins called histones, into a complex material called chromatin. In a non-dividing cell, chromatin appears as a diffuse network of thin threads and granules.
DNA plus proteins is chromatin.
Correct. Then, during the process of cell division, this chromatin becomes progressively more tightly coiled and condensed, eventually becoming visible under a light microscope as distinct, recognizable structures, the chromosomes. The number of chromosomes is characteristic for each species, though it varies hugely across the plant kingdom, from just a few in some species to over 1,000 in some ferns.
Wow, over 1,000.
Yeah. And most body cells, somatic cells, in plants, like in humans, are diploid, meaning they have 2 sets of chromosomes, one inherited from each parent, often symbolized as toucans. The reproductive cells, or gametes, like sperm and egg cells, are haploid, having only 1 set. Some plants are even polyploid, meaning they have more than 2 sets of chromosomes, which can sometimes lead to larger cells or other unique traits.
Diploid, haploid, polyploid. Okay. And what about the nucleolus that's often seen inside the nucleus? What's its specific job?
The nucleolus, or sometimes multiple nucleoli. It's usually a dense, roughly spherical-looking structure within the nucleus, noticeably darker than the surrounding chromatin. It's particularly rich in RNA and proteins, and it also contains specific loops of DNA from certain chromosomes that carry the genes for making ribosomal RNA. Its main job is essentially being the cell's ribosome factory.
Ribosome factory.
Yes, this is the primary site where ribosomal RNA, a key component of ribosomes, is synthesized based on those DNA templates. And it's also where this newly made RNA is assembled together with ribosomal proteins, which are actually synthesized in the cytoplasm and imported into the nucleus to form the large and small subunits of ribosomes. These subunits are then exported back out to the cytoplasm through the nuclear pores, where they will join together to form functional ribosomes.
So the nucleolus makes the ribosome parts inside the nucleus, but where does the actual protein building happen using those ribosomes?
That vital job happens outside the nucleus in the cytoplasm carried out by the assembled ribosomes. Ribosomes themselves are tiny, complex particles made of both rRNA, synthesized in the nucleolus, and proteins. They are the actual sites where the genetic information carried from the nucleus by messenger RNA, mRNA, is translated into the specific sequence of amino acids that makes up a polypeptide chain.
Where do you find them in the cytoplasm?
They're found in two main locations. Some are free ribosomes just floating in the cytosol. These typically synthesize proteins that will function within the cytosol itself, or perhaps be imported into the nucleus, mitochondria, or plastids. Other ribosomes are attached ribosomes bound to the surface of the endoplasmic reticulum membrane system, or sometimes to the outer membrane of the nuclear envelope. These attached ribosomes generally synthesize proteins that are destined either to be inserted into membranes or to be packaged within organelles like the vacuole, or to be secreted outside the cell entirely.
Free or attached, depending on the protein's destination.
Exactly. And when a cell is actively synthesizing a lot of a particular protein, you often see multiple ribosomes translating the same mRNA molecule simultaneously. These clusters of ribosomes working together on a single mRNA strand are called polysomes, or polyribosomes. They look like beads on a string and are very efficient little protein assembly lines. Oh, and just to reiterate that point about organelles, plastids, and mitochondria also have their own smaller ribosomes inside them, which are structurally quite similar to the ribosomes found in prokaryotes bacteria, further supporting that endosymbiotic origin theory.
Right, the ribosomes inside them look bacterial. Okay. Now let's talk about those truly distinctively plant organelles, the plastids. What's the big picture role for these, and why are they so special to plants?
Plastids really are characteristic components of plant cells, and algae too. They're primarily concerned with critical processes like photosynthesis, capturing light energy in various forms of storage like starch or oils, or synthesizing important molecules like pigments. The big picture inside, I think, is that they represent these incredibly adaptable, multi-purpose compartments within the plant cell. They likely evolved from ancient symbiotic bacteria, and they give plants unique capabilities, especially the ability to make their own food from sunlight.
Adaptable and essential for making food. What's their basic structure?
Every mature plastid is enclosed by an envelope consisting of two concentric membranes, an outer and an inner membrane. Inside this envelope, there's a complex internal structure. Generally, they feature an internal system of flattened, interconnected sacs called thylakoids. These thylakoids are often arranged in stacks, and they're surrounded by a more fluid or gel-like matrix called the stroma.
Double membrane, thylakoids, stroma, got it. And there are different types.
Yes. There are three main types found in mature plant cells, and they showcase this remarkable flexibility and specialization. The most famous, of course, are the chloroplasts.
The green ones.
The green ones. These are the undisputed sites of photosynthesis. Their green color comes from the chlorophyll pigments they contain, which are essential for capturing light energy. They also contain other pigments called carotenoids, which are typically yellow or orange, but their color is usually masked by the much more abundant chlorophylls.
Where are the pigments located?
The chlorophylls and carotenoids are embedded right within the membranes of those internal thylakoid sacs we mentioned. This location is perfect for capturing light energy and initiating the photosynthetic process. Chloroplasts are typically disc-shaped or lens-shaped, and they can be quite numerous in the cells of green tissues, especially leaves, maybe 20 to 50 per cell, sometimes more.
And they just sit there.
Oh no, they're quite dynamic. Chloroplasts can actually reorient themselves within the cell in response to light intensity. In dim light, they might spread out parallel to the cell wall to maximize light absorption. But in really bright, potentially damaging light, they might move and orient themselves perpendicularly to the light rays, sort of edge on, to minimize absorption and prevent damage. It's quite clever.
They move to adjust to the light. Cool.
Inside the chloroplast, those thylakoids are often organized into stacks that look like piles of coins. These stacks are called grana, singular granum. The individual thylakoids within a granum are called grana thylakoids, and they are interconnected by other unstacked thylakoids that traverse the stroma, called stroma thylakoids. It's a highly organized internal membrane system designed to maximize the efficiency of photosynthesis.
Okay, so they're the powerhouses capturing sunlight. But do they also store stuff? And what about that unique origin story you hinted at?
Yes, they often do store things temporarily. Chloroplasts frequently contain visible starch grains, especially when the plant is actively photosynthesizing faster than the sugars can be exported. This starch serves as a temporary reserve of the energy captured. They can also contain small lipid droplets.
And the origin.
Right. Chloroplasts are considered semi-autonomous organelles. This means they possess their own machinery for replicating themselves and synthesizing some of their own proteins. They have their own circular DNA molecules, much like bacterial chromosomes, located in clear areas of the stroma called nucleoids. They also have their own small ribosomes, which, as we noted, resemble bacterial ribosomes. And remarkably, chloroplasts replicate not through mitosis, but by a process called binary fission, essentially splitting in two, just like bacteria do.
DNA, ribosomes, splitting like bacteria.
Exactly. All this provides incredibly strong evidence for the endosymbiotic theory. The idea is that chloroplasts originated billions of years ago from free-living photosynthetic bacteria, specifically cyanobacteria, that were engulfed by an early non-photosynthetic eukaryotic cell. Instead of being digested, they established a symbiotic relationship, with the bacterium providing food through photosynthesis and the host cell providing protection and resources. Over evolutionary time, most of the bacterial genes migrated to the host cell's nucleus, but the chloroplast retains some of its own DNA and machinery. They are, after all, the ultimate source of virtually all our food supplies and most of our fuel.
Wow, bacteria living inside cells became chloroplasts. Amazing. What about the other types of plastids?
Right, besides chloroplasts, there are two other main types. First, chromoplasts. The name means colored plastid. These are pigmented plastids, but they generally lack chlorophyll. Instead, they specialize in synthesizing and retaining large amounts of other pigments, specifically the carotenoids, the yellows, oranges, and reds.
Where do you find chromoplasts?
They are responsible for the vibrant colors of many flowers, like daffodils, ripening fruits, like tomatoes turning red or bell peppers turning yellow or orange, and even the spectacular colors of some autumn leaves after the chlorophyll breaks down. They play an essential role as visual attractants for animals involved in pollination or seed dispersal. They often develop from chloroplasts as the chlorophyll degrades and carotenoid synthesis ramps up.
So green chloroplasts can become colorful chromoplasts.
Yes, they can. And the third main type are the leucoplasts. Leuco means white or colorless. These are non-pigmented plastids, and they are often structurally less complex internally than chloroplasts or chromoplasts. Their primary role is typically storage.
Storing what?
Some leucoclasts, called amyloplasts, amylo refers to starch, specialize in synthesizing and storing large quantities of starch, particularly in storage organs like potato tubers, roots like carrots, or seeds like grains. They use glucose shipped into them to build starch polymers. Other types of leucoplasts might be involved in synthesizing oils or proteins.
Starch storage specialists. Okay, and you mentioned they can transform. This flexibility sounds really important.
Absolutely. This remarkable developmental flexibility is a key feature of plastids, making them highly adaptable to the plant's needs and environmental conditions. It all starts with small, undifferentiated plastids called proplastids found in actively dividing cells, like those in embryos or shoot tips. These proplastids are the precursors and can develop into any of the mature plastid types, chloroplasts, chromoplasts, or leucoplasts, depending on the cell type and the environmental signals.
So a proplastid can become any of them.
Yes. For example, if a plant seedling starts growing in the dark, its proplastids don't develop into fully functional chloroplasts right away. Instead, they form an intermediate stage called an adioplast. Adioplasts lack chlorophyll but contain a characteristic internal structure of membranes called a prolamellar body. As soon as you expose that seedling to light, these adioplasts rapidly transform. The prolamellar body disperses, thylakoids form, chlorophyll is synthesized, and they become functional chloroplasts within hours.
Light triggers the change.
Exactly. And chloroplasts can change into chromoplasts during fruit ripening, as we said. Or leucoplasts might develop into chloroplasts if, say, a potato tuber is exposed to light, which is why potatoes can turn green. This interconversion allows plants to adapt efficiently to changing conditions and developmental needs. It's an incredibly efficient use of cellular resources.
That plasticity is amazing. Okay, moving on from plastids. If chloroplasts capture sunlight energy, then mitochondria. They're the ones burning the fuel to release that energy, right? And what's their connection to that endosymbiotic story? Are they similar to chloroplasts in origin?
That's a very good way to think of it, yes. Mitochondria, singular mitochondria, are often called the powerhouses of the cell. Like plastids, they're also enclosed by a double membrane, an outer membrane, and an inner membrane. However, the intermitochondrial membrane is highly folded into numerous intricate structures called cristae. These folds dramatically increase the surface area of the inner membrane.
Why is more surface area important?
Because that inner membrane is where the key energy-releasing reactions of the later stages of cellular respiration take place. Mitochondria are the primary sites where the chemical energy stored in organic molecules, primarily glucose derived directly or indirectly from photosynthesis, is released through oxidation and efficiently converted into the readily usable energy currency of the cell, ATP. So more surface area means more space for the protein machinery that makes ATP.
The main ATP factories.
Exactly. Plant cells, like animal cells, typically contain many mitochondria, maybe hundreds or even thousands per cell, depending on the cell's metabolic activity and energy demand. Cells that do a lot of work, like transporting substances, will have more mitochondria. Besides ATP production, they're also involved in synthesizing other important molecules like certain amino acids and vitamins.
And the endosymbiosis connection.
Yes, just like chloroplasts, mitochondria are prime examples of semi-autonomous organelles, with incredibly strong evidence pointing to an endosymbiotic origin. Like bacteria, they contain their own genetic system. They have their own circular DNA molecules housed in nucleoids within their internal matrix. They also have their own small ribosomes, again, structurally similar to bacterial ribosomes, for synthesizing some of their own proteins. And just like chloroplasts and bacteria, mitochondria also replicate primarily by binary fission, dividing independently of the cell's nuclear division cycle.
So same lines of evidence as for chloroplasts.
Precisely. The widely accepted endosymbiotic theory proposes that mitochondria originated from ancient, free-living bacteria specifically, bacteria that were very efficient at carrying out aerobic respiration, using oxygen to break down fuel. These bacteria were engulfed by larger, perhaps anaerobic, host cells. A symbiotic relationship formed where the engulfed bacterium provided large amounts of ATP to the host, and the host provided nutrients and protection. Over evolutionary time, this partnership became permanent, leading to the mitochondria we see in virtually all eukaryotic cells today. This event is thought to have been a major step in eukaryotic evolution, providing cells with a massive energetic advantage that allowed for greater complexity. And one more fascinating role for mitochondria, especially relevant in multicellular organisms. They play a pivotal role in initiating programmed cell death, or apoptosis.
They trigger cell death.
Yes. Under certain developmental signals or stress conditions, mitochondria can swell and release specific molecules, like cytochrome C, into the cytosol. This release acts as the trigger that activates a cascade of enzymes, leading to the cell's orderly self-destruction. This process is essential for normal development, like shaping fingers and toes, and for defense against disease, like eliminating infected cells. They're also constantly moving within the cell, fusing together, dividing, and often congregating in areas where energy demand is highest, like near the plasma membrane during active transport.
Wow, much more than just powerhouses. Okay, what about peroxisomes? I don't hear as much about these smaller organelles. What do they do? Are they also from endosymbiosis?
Peroxisomes, sometimes also called microbodies, are generally smaller, roughly spherical organelles, and they're enclosed by only a single membrane, unlike the double membranes of mitochondria and plastids. Their interior often appears granular and may contain a dense, sometimes crystalline core of protein.
Single membrane. And their origin?
Their origin is a bit different. While they were once thought to possibly bud off from the ER, current evidence suggests they're actually self-replicating. Much like mitochondria and plastids, they grow and divide. However, unlike those other two, peroxisomes lack their own DNA or ribosomes. This means they must import all of their necessary proteins from the cytosol, synthesized under the direction of nuclear genes. So, no direct endosymbiotic origin like the others. They seem to be quite dynamic, moving around the cell, sometimes closely associated with other organelles like chloroplasts and mitochondria.
Okay, self-replicating but need parts imported. What are their functions?
They carry out a variety of specialized metabolic tasks, often involving reactions that produce or consume hydrogen peroxide, H2O2, which is where they get their name. Peroxisomes contain enzymes like catalase that can safely break down this potentially toxic hydrogen peroxide. In plants, some peroxisomes found in green leaves play a key role in a process called photorespiration.
Photorespiration. Sounds like photosynthesis in reverse.
It kind of is, in a way. It's a metabolic pathway that occurs in the light, consumes oxygen, and releases carbon dioxide, effectively undoing some of the work of photosynthesis. It's often considered metabolically wasteful, but it might have protective roles. Peroxisomes work closely with chloroplasts and mitochondria during photorespiration.
Okay. Any other types?
Yes, there's another important type of specialized peroxisome found in plants, particularly in the fat-storing tissues of germinating seeds. These are called glyoxysomes. They contain the specific enzymes needed to convert the stored fats, lipids, into carbohydrates, specifically sucrose. This sucrose can then be transported to the growing parts of the seedling and used as an energy source and for building materials before the seedling can establish its own photosynthesis.
So fat to sugar conversion for seeds, that's crucial.
Absolutely vital for getting the seedling started. And interestingly, these two types, the leaf peroxisomes involved in photorespiration and the seed glyoxysomes, are often interconvertible, again showing that plant cell adaptability.
Adaptability again. Okay, now for the vacuole, that huge liquid-filled sac. It's one of those defining plant features, you said, taking up maybe 90% of the volume. What's its biggest contribution to a plant's life, being so large?
Absolutely. The large central vacuole, along with plastids in the cell wall, is one of the three characteristic structures that truly distinguish most mature plant cells. And yes, in many mature plant cells, this single large vacuole can indeed occupy up to 80 or 90% of the total cell volume, pushing the cytoplasm and nucleus into a thin layer pressed against the cell wall. It's closed by that single membrane, the tonoplast, which carefully controls what moves between the cytosol and the vacuole interior.
Why be so big? Is it just cheap filler?
That's actually a key part of its contribution. It's an incredibly efficient strategy for increasing cell size. By filling so much volume with relatively inexpensive cell sap, mostly water with dissolved solutes, the plant cell can achieve a large size without having to synthesize huge amounts of expensive, nitrogen-rich cytoplasmic material like proteins and nucleic acids. This large size also creates a large surface area to volume ratio for the cytoplasm itself, facilitating exchange with the environment across the plasma membrane. So much of the actual increase in the size of a growing plant cell results simply from the enlargement of its vacuole as they take up water.
Smart strategy for growth. So it's mostly water in that cell sap, but what else is in there? And what does this big central vacuole actually do for the plant, besides taking up space?
The cell sap inside the vacuole is primarily water, yes, but it's far from pure water. It's actually a complex aqueous solution containing a wide variety of dissolved substances. These can include inorganic ions like potassium, calcium chloride, sugars, organic acids, which often make the sap acidic, and amino acids. Sometimes the concentrations of certain substances can be so high that they precipitate out and form visible crystals within the vacuole, like crystals of calcium oxalate, which are common in many plant cells. Vacuoles don't typically synthesize these molecules themselves. They actively transport them in from the surrounding cytoplasm via proteins embedded in the tonoplast membrane.
Okay, a watery storage solution. What are its main functions then?
Its major functions are incredibly diverse and absolutely vital for plant life. You can list several key roles. First, and perhaps most importantly for plant structure, the vacuole is crucial for maintaining turgor pressure.
Turgor pressure.
Yes. Water tends to move into the vacuole by osmosis because of the high concentration of solutes inside. As the vacuole swells with water, it pushes the cytoplasm firmly against the rigid cell wall. This internal pressure, called turgor pressure, is what keeps plant cells firm, or turgid. It's essential for maintaining the rigidity of non-woody plant tissues, like leaves and stems, allowing them to stand upright and hold their shape. If a plant loses too much water, the vacuoles shrink, turgor pressure drops, and the plant wilts.
Ah, so turgor is why plants don't flop over.
Largely, yes, for herbaceous plants. Second, vacuoles act as important storage compartments. They can store reserves of primary metabolites like proteins, especially in seeds, or sugars. But critically, they also serve as a place to sequester potentially toxic secondary metabolites, things like nicotine, tannins, alkaloids, isolating them safely away from the sensitive metabolic machinery in the cytoplasm where they could cause harm to the plant itself. This is a key defense mechanism against herbivores, as the toxins are released only when the cell is broken open by chewing.
Storing toxins safely away. Clever.
Third, the vacuole is often a site of pigment deposition, contributing directly to plant coloration. Those brilliant blue, violet, purple, dark red, and scarlet colors you see in many flowers, like pansies, petunias, fruits like grapes, and vegetables like beets or red onions, are frequently due to water-soluble pigments called anthocyanins, dissolved directly in the cell sap within the vacuole.
Anthocyanins again.
Yes, and here's that beautiful aha moment connecting back to autumn leaves. Anthocyanins are also responsible for many of the spectacular red and purple colors of autumn foliage. These pigments are actively synthesized in the leaves of some species in response to environmental cues like cool temperatures and bright sunlight in the fall. As the green chlorophyll in the chloroplast degrades and disappears, these newly formed red-purple anthocyanins stored in the vacuoles are unmasked, revealing their vibrant hues against the yellows and oranges from carotenoids, which were already in the plastids.
So fall colors are a mix of unmasking old pigments and making new ones in the vacuole.
Often, yes. And finally, vacuoles also have a role in degradation and recycling. They contain various hydrolytic enzymes, similar to those found in animal cell lysosomes. They are involved in breaking down macromolecules, waste products, and even entire organelles like old mitochondria or plastids that are no longer functional. The breakdown products can then be recycled back into the cytoplasm for reuse by the cell. So they're definitely not just inert storage bags. They are dynamic, multifunctional compartments absolutely essential for the plant cell's life, structure, defense, and adaptability.
Dynamic and multitasking. Got it. Okay, next up on our tour, the endoplasmic reticulum, the ER. This sounds like a cellular highway system and maybe a factory all rolled into one. What's its role in the cell's production line and transport network?
That's a really good description. The endoplasmic reticulum, ER, is a truly extensive and complex three-dimensional membrane network that permeates throughout the entire cytosol of eukaryotic cells. It forms a network of interconnected flattened sacs, called cisternae, and tubules. It essentially divides the cytoplasm into compartments and provides a huge surface area for biochemical reactions. Its form and abundance can vary greatly depending on the cell type and its metabolic activity.
And there are two types, right? Rough and smooth.
Exactly. We recognize two main types, which are often interconnected but have different structures and primary functions. First, there's the rough ER. This form gets its name because its outer surface, the side facing the cytosol, is studded with numerous ribosomes, giving it a rough appearance under an electron microscope. Rough ER often consists primarily of flattened sacs or cisternae, cells that are specialized for synthesizing and secreting a lot of proteins, or for storing proteins, like in seeds, tend to have abundant rough ER. Its main role is in the synthesis, folding, modification, and transport of proteins that are destined for insertion into membranes or delivery to certain organelles like the Golgi or vacuoles or secretion out of the cell.
So protein factory and modification center.
Largely, yes. Then there's the smooth ER. This form lacks ribosomes on its surface, hence it looks smooth. It's often more tubular in structure than the rough ER. Its primary role is in lipid synthesis, including synthesizing oils, phospholipids from membranes, and steroids. Cells that secrete lipids, like those making waxes for the cuticle, tend to have extensive smooth ER. It's also involved in other functions like detoxification in some cells.
Lipid factory. And they're connected.
Yes, both rough and smooth ER are typically interconnected within the same cell, forming one continuous network. The relative amounts of each can change depending on the cell's activities. And interestingly, the outer membrane of the nuclear envelope, which surrounds the nucleus, is often physically continuous with the rough ER membrane, linking these two major membrane systems. In fact, the nuclear envelope actually breaks down into ER-like fragments during mitosis and then reforms from ER vesicles afterwards.
So it's a synthesis factory and a transport network.
Exactly. It functions as a crucial communication and transport system within the cell, channeling materials like newly synthesized proteins and lipids to different parts of the cell, often towards the Golgi apparatus for further processing. There's also a specialized network of ER called the cortical ER, located just inside the plasma membrane. This cortical ER seems to play roles in regulating calcium ion levels in the cytosol, which is important for signaling, and it also interacts with the cytoskeleton and plasma membrane, perhaps anchoring structures.
And it makes lipids. What about those oil bodies in seeds?
Ah, good connection. The ER is indeed one of the main sites of lipid synthesis in plants. And those oil bodies, or lipid droplets, which store triglycerides, especially in seeds, actually arise directly from the ER membrane. They seem to bud off from the ER before being released into the cytosol. They are unique in that they are surrounded by a single monolayer of phospholipids, not a bilayer, embedded with specific proteins. These oil bodies are incredibly important energy reserves for the developing seedling upon germination.
Budding off the ER. Cool. And the ER is connected to other cells, too. You mentioned plasmodesmata.
Yes, absolutely, and this is really critical for plant function. The ER membranes, particularly the cortical ER of adjacent plant cells, are directly interconnected through those plasmodesmata, the cytoplasmic channels that run through the cell walls. A tube of ER, called the desmotubule, actually passes through the center of each plasmodesma. This provides a direct, continuous pathway for the ER system throughout the entire plant body. This likely facilitates rapid cell-to-cell communication and transport of certain molecules, reinforcing that idea of the plant functioning as a unified organism, the symplast.
Wow, an ER network connecting the whole plant. Okay. If the ER is the factory, then the next stop, the Golgi apparatus, sounds like it should be quality control and the shipping department. How does it process and deliver molecules?
That's a perfect analogy. The Golgi apparatus, sometimes called the Golgi complex or Golgi body, or in plants, often referred to as a dictyosome for a single stack, acts very much like a central processing and packaging center in the cell's endomembrane system.
What does it look like?
It typically consists of a stack of flattened, membrane-bound sacs called cisternae. Think of a stack of slightly curved pancakes or pita breads. Unlike in many animal cells, where the Golgi might be one large complex near the nucleus, plant cells often have many separate, smaller Golgi stacks scattered throughout the cytoplasm. And these plant Golgi stacks usually remain intact and active even during cell division.
Okay, stacks of sacs. How does the packaging work? Is it directional?
Yes, the Golgi is a dynamic and highly polarized structure. It has this distinct entry face called the cis face or forming face, which is usually oriented towards the ER. And it has an exit face called the trans face or maturing face, which faces away from the ER, often towards the plasma membrane. Materials like proteins and lipids synthesized in the ER typically enter the Golgi at the cis face, move progressively through the stack of cisternae from cis to trans, undergoing modifications along the way, and then exit from the trans face, often from a complex network of tubules and vesicles called the trans-Golgi network, TGN.
Processing as they move through the stack. What kind of processing?
All sorts. The Golgi is heavily involved in modifying, sorting, and packaging proteins and lipids it receives from the ER. For example, glycoproteins, proteins with attached carbohydrates, might have their sugar chains modified or further elaborated as they pass through the Golgi cisternae. In plants, the Golgi has a particularly crucial role in synthesizing certain complex polysaccharides.
Like for the cell wall.
Exactly. While cellulose is made at the plasma membrane, the Golgi is the site where most of the non-cellulosic polysaccharides of the cell wall matrix, things like hemicelluloses and pectins, are synthesized. These are then packaged into vesicles for delivery to the cell wall. This synthesis happens in a stepwise fashion as the molecules move across the Golgi stacks. It also processes and sorts proteins received from the rough ER, sending them to their correct final destinations, maybe to the vacuole or lysosomes in animal cells, or packaging them for secretion outside the cell. Think of it as a series of conveyor belts in a factory, each station performing a specific modification or sorting step on the cargo as it moves along.
Okay, modifying, sorting, packaging. And how do these packaged substances actually leave the Golgi and get to where they need to go?
These newly processed and sorted substances are packaged into small, membrane-bound sacs called vesicles that bud off from the trans face or the TGN. The type of vesicle can depend on the cargo and destination. For example, proteins destined for the vacuole might be packaged into coated vesicles, which have a specific protein coat, like clathrin, on their outer surface that helps target them correctly. Substances destined for the cell wall, like those pectins and hemicelluloses, are usually packaged into non-coated secretory vesicles.
And then the vesicles travel.
Yes, these secretory vesicles, often guided along tracks of the cytoskeleton, like actin filaments, then migrate to the plasma membrane. They fuse with the plasma membrane and release their contents outside the cell, a process called exocytosis. The vesicle membrane itself becomes incorporated into the plasma membrane, contributing to its growth. Conversely, cells can also take up substances from outside by forming vesicles at the plasma membrane that pinch inward, a process called endocytosis.
Exocytosis out, endocytosis in. So the ER, Golgi, nuclear envelope, vesicles, and plasma membrane, they're all connected in a way, part of one big system.
Yes, exactly. This brings us to the concept of the endomembrane system. It's generally considered to be this interconnected network of most of the cell's internal membranes, including the nuclear envelope, the ER, rough and smooth, the Golgi apparatus, lysosomes in animals, vacuoles in plants, vesicles, and the plasma membrane. It excludes the membranes of mitochondria, plastids, and peroxisomes, which have different origins and functions. The ER is seen as the initial source of lipids and many membrane proteins. These components are then transported, often via vesicles, to the Golgi, where they are modified, sorted, and then used to form the membranes of other organelles within the system or are secreted. It's a continuous dynamic system for synthesizing, modifying, and transporting membranes and the substances they contain throughout the eukaryotic cell. It's all about flow and processing.
A dynamic flow of membranes and materials.
Okay. Now, every structure we've discussed so far, all these organelles moving around, the cell changing shape, it all needs some kind of internal support structure and a way to actively move things. That's where the cytoskeleton comes in, right? What are its main components and roles in a plant cell?
Precisely. You can't have all this complex organization and dynamic activity without an internal framework. The cytoskeleton is exactly that. It's a complex, dynamic, three-dimensional network of protein filaments that extends throughout the cytosol of all eukaryotic cells. It's crucial for maintaining cell shape, providing mechanical support, enabling cell movement in some cases, anchoring organelles in place, directing the movement of organelles and vesicles within the cell, and it plays absolutely essential roles in cell division, growth, and differentiation. In plant cells, the cytoskeleton consists mainly of two major types of protein filaments, microtubules and actin filaments, also known as microfilaments.
Okay, let's take microtubules first. What are they like? Microtubules are relatively stiff, hollow cylinders, about 24 nanometers in diameter. They're built from subunits of a globular protein called tubulin. These tubulin subunits assemble head to tail in long rows, or protofilaments, and typically 13 of these protofilaments associate side by side to form the hollow tube. Microtubules have distinct ends, a plus end and a minus end, which affects how they grow and shrink, and they exhibit a property called dynamic instability.
Dynamic instability.
Yeah, it means they are constantly undergoing periods of rapid growth, polymerization, followed by sudden shrinkage, depolymerization. They're constantly breaking down, reforming, and rearranging within the cell. Think of it like quickly assembling and disassembling a structure made of tiny, hollow building blocks. This dynamic nature allows the cytoskeleton to rapidly reorganize in response to the cell's needs. Their assembly and organization are often controlled by specific sites in the cell called microtubule organizing centers, MTOCs, although these are less defined in plant cells compared to animal cells.
Okay, dynamic hollow tubes. What are their main jobs in the plant cell?
They have many crucial roles. In growing or enlarging plant cells, there's often a network of cortical microtubules lying just inside the plasma membrane, aligned parallel to each other. These cortical microtubules play a critical role in guiding the orderly deposition of cellulose microfibrils during cell wall synthesis.
They guide the cellulose making.
Yes, they essentially act as tracks that direct the movement of the cellulose synthase enzyme complexes in the plasma membrane. Since the orientation of cellulose microfibrils determines the direction in which the cell can expand against turgor pressure, these microtubules effectively control the directionality of cell growth and thus influence the overall shape of the plant organ.
Wow, controlling plant shape at the micro level. What else?
They also help direct the movement of Golgi vesicles carrying cell wall materials to the plasma membrane. They form the essential framework of the spindle fibers, the mitotic spindle, that separates the chromosomes during mitosis and meiosis. And they are the core structural components of eukaryotic flagella and cilia, the whip-like appendages used for motility in some cells.
Okay, guiding growth, moving chromosomes, making flagella. What about the other type, actin filaments?
Actin filaments, which are also often called microfilaments because they're thinner than microtubules, only about 5 to 7 nanometers in diameter, are long, flexible filaments composed of subunits of another globular protein called actin. Like microtubules, they are also polar structures, having distinct ends, and can exist as single filaments, in bundles, or in networks.
Thinner and more flexible. What do?
They are involved in a really wide variety of cellular activities. In plants, they play roles in cell wall deposition, particularly in the specialized tip growth seen in pollen tubes and root hairs, where growth is highly localized at the very tip. They are involved in moving the nucleus to the correct position within the cell before and after cell division. They participate in vesicle-mediated secretion, helping to move those secretory vesicles. And critically, actin filaments are the primary drivers of cytoplasmic streaming, or cyclosis.
Ah, that internal stirring motion. How do actin filaments drive that?
That fascinating, orderly bulk flow of cytoplasm and organelles within the plant cell is primarily driven by the interaction between actin filaments and another type of protein called myosin. Myosin acts as a motor protein. Using energy from ATP hydrolysis, myosin molecules essentially walk along the actin filament tracks, dragging organelles or masses of cytoplasm along with them. This continuous movement helps distribute nutrients, metabolites, and organelles efficiently throughout the relatively large volume of many plant cells.
Motor proteins walking on actin tracks, pulling stuff along. Cool. So flagella and cilia, you mentioned microtubules are in them. Are they common in plants?
Not as common as in animals or protists. Flagella and cilia are essentially the same structure, long hair-like appendages extending from the cell surface, used for locomotion or moving fluid. We tend to call them cilia if they're short and numerous, and flagella if they're long and few. In the plant kingdom, true flagella are primarily found only in the motile reproductive cells, specifically the swimming sperm cells of bryophytes, mosses, pteridophytes, ferns and their allies, and some gymnosperms, cycads and ginkgo. Flowering plants lack flagellated sperm.
Only in the sperm of some groups.
And their structure. Each eukaryotic flagellum, or cilium, has a remarkably conserved and precise internal organization. There's an outer ring consisting of 9 pairs, doublets, of microtubules, surrounding 2 single central microtubules. This is the characteristic 9 + 2 minutes arrangement found in virtually all eukaryotic flagella and cilia. Their whip-like movement is generated by the outer microtubule doublets sliding past each other, a process powered by motor proteins, dynein arms attached between the doublets, again using ATP. They grow out from structures called basal bodies located in the cytoplasm at the base of the flagellum, which have a similar microtubule structure, but typically 9 triplets with no central pair.
Okay, the cytoskeleton provides structure, movement, and even swimming ability in some cases. Now let's talk about the cell wall again. We keep coming back to it. This is truly what makes a plant cell, right? What's its fundamental composition, and what would you say is its single most important function?
Yes, above all other characteristics, the presence of a cell wall is arguably the major distinguishing feature of plant cells. It's that relatively rigid, often quite strong outer layer that surrounds the plasma membrane. Its single most important function, I would say, is to provide structural support and limit the expansion of the protoplast. Because plant cells live in environments that are often less concentrated than their own cytosol, water tends to enter the cell by osmosis. Without the cell wall providing a counterpressure, the plasma membrane would swell and eventually burst, just like an animal cell would in pure water.
So it prevents the cell from exploding.
Exactly. It constrains the expansion caused by turgor pressure, but it does much more than that. The cell wall largely determines the final size and shape of the plant cell, and consequently the overall form and texture of the plant tissue. Think of the difference between a soft leaf and hardwood that's largely down to differences in their cell walls. And importantly, the cell wall is far from being just a passive inert box around the cell. It's metabolically active, it contains various enzymes, it's involved in absorption, transport, and secretion processes at the cell surface, and it plays a crucial role in defense against pathogens and pests.
Active and protective, too. What's it primarily made of again?
Its principal structural component, the reinforcing framework, is cellulose. As we discussed back in carbohydrates, cellulose is that polysaccharide made of beta-glucose units linked together in long, straight chains. These chains aggregate side by side to form strong, cable-like structures called cellulose microfibrils. These microfibrils have incredibly high tensile strength. They resist stretching forces very effectively, stronger than steel by weight, as we said.
Cellulose microfibrils are the rebar, basically.
That's a great analogy. They form the reinforcing framework. This cellulose framework is then embedded in and interlocked by a complex cross-linked matrix composed of other non-cellulosic molecules. The main components of this matrix are other polysaccharides, primarily hemicelluloses and pectins, along with some structural glycoproteins, proteins with carbohydrates attached.
Okay, cellulose plus a matrix of other stuff. Do these matrix components have specific jobs too?
Indeed they do. They're not just filler. They contribute significantly to the wall's properties. Hemicelluloses are branched polysaccharides that can hydrogen bond tightly to the surfaces of the cellulose microfibrils. They essentially tether the microfibrils together, limiting the wall's ability to stretch and thus helping to regulate cell enlargement. Pectins, on the other hand, are a very different group of branched polysaccharides. They are highly hydrophilic, meaning they attract and hold a lot of water, forming a gel-like consistency within the matrix. This water-filled pectin gel makes the primary wall somewhat plastic and pliable, which is essential to allow for cell growth and expansion. Pectins are also the main component of the middle lamella, that intercellular layer that glues adjacent plant cells together.
Hemicellulose for tethering, pectin for flexibility and glue. Got it. Is there anything else important in the wall?
One other polysaccharide worth mentioning is callus. Callus is interesting because it's not usually a major structural component of healthy walls, but it can be rapidly synthesized and deposited at specific sites in response to wounding or stress. For example, it's quickly laid down around plasmodesmata if a cell is damaged, effectively sealing off the connections to neighboring cells and preventing the spread of damage or pathogens. It also forms temporary structures during cell division and pollen development.
A quick sealant for wounds, okay. Are there different layers to the cell wall? Is it the same all the way through?
No, cell walls are often layered, and their structure and composition can vary greatly depending on the cell type, its age, and its function. We generally distinguish between a primary wall and sometimes a secondary wall.
Primary first.
Yes. The primary wall is the first layer formed by a developing plant cell. It's deposited before and during cell growth and expansion. It's typically relatively thin and flexible, allowing the cell to enlarge. Its composition is mainly cellulose microfibrils embedded in that matrix, rich in hemicelluloses, pectins, and some structural proteins, plus a lot of water, making it pliable. Cells that remain actively dividing or are metabolically very active, like photosynthetic cells in leaves or storage cells in fruits, often only have a primary wall throughout their lives. This flexibility is important for their function and also allows for processes like wound healing. Primary walls aren't usually uniform in thickness. They often have thin areas called primary pit fields, which are typically clusters of plasmodesmata passing through the wall.
Flexible first layer for growing cells, then the middle lamella.
Right, the middle lamella is actually the outermost layer, technically. It's that pectin-rich layer that forms between adjacent cells during cell division, effectively cementing them together. So you have middle lamella, then the primary wall of cell 1, primary wall of cell 2, middle lamella again, and so on.
Okay. And the secondary wall?
Some plant cells, especially those that need extra strength or have a specialized function like water conduction, like xylem cells, or mechanical support, like fibers in wood, deposit an additional layer inside the primary wall. This is the secondary wall, and it's typically laid down after the cell has stopped growing and enlarging.
Stronger and laid down later. How is it different?
Secondary walls generally have a much higher percentage of cellulose than primary walls, and the microfibrils are often densely packed and highly organized, sometimes in distinct layers with different orientations, which provides tremendous strength. They usually contain much less pectin and water, making them rigid and strong rather than flexible. And very importantly, secondary walls commonly become impregnated with lignin. As we discussed with phenolics, lignin is that complex polymer that adds significant compressive strength, resistance to squeezing, stiffness, and waterproofing to the wall. Wood is composed mainly of cells with thick, lignified secondary walls.
Lignin adds the real rigidity.
Absolutely. Also, secondary walls don't form over the primary pit fields. Instead, they have characteristic eruptions or gaps called pits. Pits in the secondary wall of one cell usually align precisely with pits in the adjacent cell, forming a pit pair. These pit pairs essentially act as channels, allowing for communication and transport, especially of water and xylem, across the otherwise impermeable secondary walls. There are different types, like simple pits and bordered pits, where the secondary wall arches over the pit cavity, creating a more complex structure often seen in water-conducting cells.
Pits act as channels through the secondary wall. Okay, so how does the cell wall actually grow and expand then, especially that primary wall, if it has all those strong cellulose microfibrils? It sounds like a delicate balance between being strong and being able to stretch.
It is a very complex and carefully controlled process, orchestrated by the living protoplast inside. The primary wall must somehow yield to the internal turgor pressure to allow the cell to expand, but it must do so without rupturing, and while still maintaining its structural integrity. It's like stretching a reinforced fabric. You need to loosen the connection slightly to allow stretch, but not break the reinforcing threads.
How does it loosen?
This involves both biochemical modifications of the matrix components, like pectins and hemicelluloses, and the action of specialized wall-loosening proteins, such as expansins, which seem to disrupt the hydrogen bonds between cellulose microfibrils and hemicelluloses, allowing the microfibrils to slide past each other slightly under the force of turgor pressure. Plant hormones like auxin are known to trigger this wall loosening. As the wall stretches, the protoplast synthesizes and deposits new wall material, both matrix components and new cellulose microfibrils, integrating them into the expanding wall structure, layer by layer, usually on the innermost surface.
Adding new material as it stretches. And the direction matters.
Critically, the overall direction of cell expansion is profoundly influenced by the orientation in which those new cellulose microfibrils are laid down. If the microfibrils are deposited randomly, the cell tends to expand equally in all directions, becoming spherical. However, if the microfibrils are oriented predominantly in one direction, say, wrapped around the cell like hoops on a barrel, the cell can expand much more easily perpendicular to that orientation. So hoops wrapped around the cell lead to elongation along the cell's long axis. This control over microfibril orientation is absolutely key to determining plant cell shape and thus the shape of the whole plant body.
Hoops make it elongate. That's clever. And how does the plant make and place those cellulose microfibrils so precisely where they need to go?
This is a beautiful example of intricate cellular coordination. The cellulose microfibrils themselves are synthesized right at the outer surface of the plasma membrane by large multi-protein enzyme complexes called cellulose synthase complexes. These complexes look like little rosettes embedded in the plasma membrane. They take glucose subunits, specifically UDP glucose, from the cytosol and polymerize them into cellulose chains, extruding the growing microfibril onto the outer surface of the membrane, directly into the cell wall space.
Enzymes in the membrane spitting out cellulose.
Exactly. And crucially, the movement of these cellulose synthase complexes within the fluid plasma membrane appears to be guided by the underlying cortical microtubules of the cytoskeleton, those tracks we talked about just inside the membrane. The enzyme complexes seem to follow along the paths laid down by the microtubules. So the orientation of the microtubules inside the cell dictates the orientation of the cellulose microfibrils being deposited outside the cell. It's a remarkable link between the internal cytoskeleton and the external cell wall architecture. Meanwhile, the matrix substances like hemicelluloses and pectins are synthesized inside the cell, in the Golgi, and delivered to the wall in those secretory vesicles we discussed, fusing with the plasma membrane to release their contents. The specific type and amount of matrix material delivered can also vary depending on the cell's developmental stage and needs. It's a highly regulated construction project.
Cytoskeleton guiding wall construction. Amazing coordination. Okay, one more structure related to the wall. We talked about the organismal theory and how plant cells aren't totally isolated because they're connected. That's where plasmodemata come in, right? Remind us what role these tiny connections play.
Exactly. Plasmodemata, singular, plasmodesma, are the physical basis for that interconnectedness, for the symplast. They are tiny, membrane-lined cytoplasmic strands that directly connect the protoplasts, the living contents, of adjacent plant cells, passing right through the intervening cell walls. They essentially make the cytoplasm of one cell continuous with the cytoplasm of its neighbors and potentially continuous throughout the entire plant.
Tunnels through the walls.
Pretty much. They appear as narrow channels, maybe 30 to 60 nanometers in diameter. The channel is lined by the plasma membrane, which is continuous from one cell to the next through the plasmodesma. And running through the center of most plasmodesmata is a narrow, modified tubule of endoplasmic reticulum called the desmotubule. This desmotubule connects the ER of one cell with the ER of the adjacent cell, ensuring continuity of that membrane system as well.
So cytoplasm and ER are connected. How do they form?
Many plasmodesmata are formed during cell division, when strands of ER get trapped within the developing cell plate as it forms between the two new daughter cells. However, plasmodemata can also form secondarily, developing across existing cell walls in non-dividing cells, although the mechanism for that is less well understood.
And their main function is transport.
Yes, they provide a vital pathway for the direct regulated transport of substances including water, ions, small molecules like sugars and amino acids, and even larger molecules like proteins and RNA signaling molecules directly from the cytosol of one cell to the cytosol of the next, bypassing the need to cross membranes. This allows for rapid, synchronized communication and efficient distribution of nutrients and signals throughout the plant tissue, enabling the plant to function as a coordinated, multicellular organism. They are absolutely critical.
The plant's internal communication network. Okay, so all these amazing structures are built, maintained, they function. But cells don't last forever. They need to divide to allow the plant to grow or replace damaged tissues. What's this whole dance of cell division called, and why does it need to be so precisely orchestrated?
That whole process is encompassed by the cell cycle. It's a regular, repeated sequence of events: growth, DNA replication, preparation, and finally division that results in one cell dividing into two, usually genetically identical daughter cells. In single-celled organisms, the cell cycle is how the population increases. In multicellular organisms like plants, it's the fundamental process that drives growth from an embryo, enables the repair of damaged tissues, and allows for the replacement of old or worn-out cells.
Growth, repair, replacement.
Right. And the absolutely critical aspect, the reason it needs to be so precisely orchestrated, is that each new daughter cell must receive a complete and exact replica of the parent cell's genetic information, the chromosomes. It also needs a sufficient share of the cytoplasm and organelles to be able to function independently. This ensures that the daughter cells are structurally and functionally similar to the parent cell, maintaining the integrity of the tissue and the organism. Before a cell can possibly divide, all of its DNA must be faithfully replicated, and it must produce sufficient quantities of organelles and other cytoplasmic components to furnish two new cells.
So it's not just about splitting in two then. There's a lot of preparation involved before the actual division happens.
Oh, absolutely. The actual division part is often relatively short compared to the preparation time. The eukaryotic cell cycle is typically divided into two main overlapping stages. There's M phase, which includes mitosis, the division of the nucleus and its duplicated chromosomes, and cytokinesis, the division of the cytoplasm. And then there's interphase, which is the period between successive M phases. Interphase is when the cell grows, carries out its normal metabolic functions, and crucially, prepares for the next division by replicating its DNA and synthesizing necessary proteins and organelles. Interphase itself is usually subdivided into three distinct phases: G1, S, and G2.
Okay, M phase is division. Interphase is preparation. Let's break down interphase. G1, S, G2.
Right, G1 phase, gap 1. This phase follows the previous M phase and precedes DNA replication. It's typically a period of intense biochemical activity and growth. The cell roughly doubles in size, synthesizes more enzymes, ribosomes, organelles like mitochondria and plastids, and membrane systems. It's basically gearing up for DNA synthesis and gathering resources. S phase, synthesis phase. This is the crucial phase where DNA replication occurs. The cell duplicates its entire nuclear genome so that each chromosome, which consisted of a single DNA molecule, chromatid in G1, now consists of two identical sister chromatids joined together at the centromere. Associated proteins, like histones that help package the DNA, are also synthesized during S phase, G2 phase, gap 2. This phase follows S phase and precedes the next M phase. Its primary role seems to be as a checkpoint and final preparation stage. The cell ensures that chromosome replication is complete and checks for any DNA damage that might need repair before committing to the complex process of mitosis. It also synthesizes proteins specifically required for mitosis, like tubulin for the spindle fibers.
G1 growth, S DNA replication, G2 final check. And there are checkpoints.
Yes, throughout this cycle there are critical checkpoints, particularly at the transitions between G1 and S and between G2 and M. These act like quality control points, monitoring the cell's condition and ensuring that all necessary events like DNA replication or repair have been properly completed before allowing the cycle to proceed to the next stage. These checkpoints are often regulated by specialized enzymes called cyclin-dependent kinases, CDKs, interacting with regulatory proteins called cyclins. If conditions aren't right, the checkpoints can halt the cycle until the problem is fixed. Oh, and speaking of DNA replication, there's a fascinating variation sometimes seen in plants called endor duplication.
Endor duplication, what's that?
It's a process where some plant cells undergo multiple rounds of DNA replication, S phase, without subsequently going through meiosis, M phase. This results in cells with gigantic nuclei, containing many copies of each chromosome. They become highly polyploid. This is thought to be a way to dramatically increase the number of gene copies, allowing for very high levels of gene expression and protein synthesis, perhaps needed for specialized functions. You see it, for example, in the cells that form the epidermal hairs, trichomes, on the leaves of the model plant Arabidopsis thaliana.
Multiple DNA copies without dividing. Interesting strategy. Now, are there any unique things that happen during plant interphase that maybe give us a clue about where the division itself is going to happen later?
Yes, there are actually two very interesting and quite predictive events that often occur during interphase in plant cells, particularly before mitosis. First, in many plant cells, especially those that have a large central vacuole, the nucleus often migrates from its position near the cell periphery to the center of the cell just before division. It becomes anchored there by cytoplasmic strands. These strands then broaden and merge to form a transverse sheet of cytoplasm containing the nucleus that bisects the large vacuole. This structure is called the phragmosome. The key thing about the phragmosome is that it forms precisely in the plane where the cell will ultimately divide later during cytokinesis.
So the nucleus moves to the middle, predicting the division plane. What's the second event?
The second event is the formation of the preprophase band. Just before the cell actually enters prophase, the first stage of mitosis, a dense, narrow, ring-like band of microtubules appears in the cytoplasm, just beneath the plasma membrane. This band encircles the nucleus in the equatorial plane, the plane where the phragmosome formed.
A ring of microtubules.
Exactly. And what's remarkable is that this preprophase band is a transient structure. It disappears completely before metaphase, long before the new cell wall actually forms. Yet it serves as an incredibly accurate marker, a memory for the cell. It precisely predicts both the future orientation of the mitotic spindle and the exact location where the new cell wall, the cell plate, will eventually fuse with the parent cell wall during cytokinesis. It's like the cell draws a precise line on the floor, marking exactly where the new wall needs to end up, even though the line gets erased before construction finishes.
That's incredible foresight by the cell. Okay,
Great.
the preparation is done, the location is marked. Now for the main event, mitosis and cytokinesis, the actual splitting of the nucleus in the cell. Can you walk us through the major steps of mitosis first?
Right. Mitosis, the division of the nucleus, is a continuous dynamic process, but for convenience, we divide it into 4 major phases based on the appearance and behavior of the chromosomes: prophase, metaphase, anaphase, and telophase. Remember PM8.
PMAU. Okay, prophase.
Prophase is typically the longest phase of mitosis. The key event here is that the diffuse chromatin, which was spread throughout the nucleus during interphase, begins to coil up, condense, and become progressively shorter and thicker. Eventually, it becomes visible under a light microscope as distinct individual chromosomes. Remember, because DNA replication happened during S phase, each of these visible chromosomes actually consists of 2 identical sister chromatids, which are still tightly joined together, especially at a constricted region called the centromere. Also during prophase, the nucleolus usually disappears, and the nuclear envelope breaks down and disperses into small vesicles, likely absorbed into the ER system.
Chromosomes condense, nucleus breaks down. Next, metaphase.
Metaphase. With the nuclear envelope gone, the mitotic spindle begins to form in the region where the nucleus used to be. The spindle is a complex, 3-dimensional structure made primarily of bundles of microtubules that stretch between 2 opposite poles of the cell. Meanwhile, specialized protein complexes called kinetochores develop on both sides of the centromere of each chromosome, 1 kinetochore per sister chromatid. Certain spindle microtubules, called kinetochore microtubules, attach to these kinetochores. These microtubules, along with other polar microtubules that extend from pole to pole and overlap in the middle, they work together to maneuver all the duplicated chromosomes, pushing and pulling them until they are all precisely aligned along the equatorial plane, also called the metaphase plate, right in the center of the cell, halfway between the 2 spindle poles.
All chromosomes lined up in the middle, then anaphase.
Anaphase is usually the shortest phase. It's very dramatic. It begins abruptly when the connections holding the sister chromatids together at their centromeres suddenly break. The sister chromatids separate simultaneously, and each one is now considered an individual daughter chromosome. These newly separated daughter chromosomes are then rapidly pulled towards opposite poles of the spindle.
How are they pulled?
It seems to be a combination of 2 things. The kinetochore microtubules attached to the chromosomes shorten, likely by losing tubulin subunits at the kinetochore end, effectively reeling the chromosomes in towards the poles. At the same time, motor proteins associated with the kinetochores may actively walk the chromosomes along the microtubules towards the poles. The polar microtubules also seem to push the poles further apart, helping to separate the 2 sets of chromosomes.
Chromatids separate and move to opposite ends. Finally, telophase.
Telophase. This phase essentially reverses the events of prophase. The 2 complete identical sets of daughter chromosomes arise at opposite poles of the cell. The spindle apparatus begins to disappear. New nuclear envelopes start to form around each set of chromosomes, reforming likely from vesicles derived from the ER. The condensed chromosomes begin to elongate, uncoil, and become indistinct again, returning to their diffuse chromatin state. Nucleoli also reappear within each newly forming daughter nucleus. At the end of telophase, mitosis, the division of the nucleus, is complete. You now have 2 genetically identical daughter nuclei within the same cell for a short time.
2 identical nuclei. Okay, mitosis is done. But how does the rest of the plant cell divide the cytoplasm, the organelles, and forming that new wall between them, that cytokinesis, right? How does it happen in plants?
Right, so cytokinesis, the division of the cytoplasm, typically begins sometime during late anaphase or telophase, while the nuclear events are still finishing up. And the way it happens in plants, and some algae, is quite distinct from how it usually occurs in animal cells, which typically pinch in from the outside. Plant cytokinesis involves the formation of a completely new structure called the cell plate, which starts forming in the middle of the cell and grows outward towards the existing cell walls.
Starts in the middle. How?
In early telophase, as the 2 sets of chromosomes are separating, a structure called the phragmoplast begins to form in the region between the 2 newly forming daughter nuclei on the former equatorial plane. The phragmoplast is a complex, barrel-shaped system composed initially of short microtubules, derived from the remnants of the polar spindle microtubules, oriented perpendicular to the eventual plane of division. along with actin filaments and associated proteins.
Okay, a phragmoplast forms in the middle. What does it do?
The phragmoplast acts as a scaffold and guide. It directs the movement of numerous small, membrane-bound vesicles, which are produced by the Golgi apparatus, to the equatorial plane. These Golgi vesicles contain the necessary polysaccharide precursors, mainly pectins and hemicelluloses, needed to build a new cell wall matrix.
Golgi vesicles arrive at the phragmoplast.
Exactly. Guided by the phragmoplast, these vesicles arrive at the center, align, and then begin to fuse together. As they fuse, they form a small, flattened membrane-enclosed disc. This is the initial cell plate. The contents of the vesicles form the matrix of the new wall, specifically the middle lamella and the beginnings of the new primary walls. And the membranes of the fusing vesicles themselves contribute to forming the new stretches of plasma membrane that will separate the two daughter cells.
So the vesicles build the plate and the new membranes.
Precisely. And here's another neat connection. Plasmodesmata, those channels connecting cells, are often formed during this process. As the Golgi vesicles fuse to form the cell plate, segments of smooth ER tubules can get trapped between them, eventually becoming the desmotubules within the newly formed plasmodesmata that will span the new wall.
Ah, plasmodesmata form as the wall builds.
Yes. The cell plate continues to grow outwards, guided by the phragmoplast, which also expands centrifugally from the center outwards, accumulating more Golgi vesicles at its edges. Eventually, the edges of the growing cell plate reach and fuse with the existing plasma membrane and cell wall of the parent cell. And where does it fuse? Precisely at the site that was previously marked by that preprophase band way back before mitosis even started.
It finds the mark.
It finds the mark. Once the cell plate is complete, it matures. A new middle lamella develops within it, cementing the two daughter cells together. Then each daughter cell deposits its own new primary cell wall on its side of the middle lamella. As the daughter cells subsequently enlarge, often by vacuole expansion, the old parent cell wall often stretches and may eventually rupture. It's an incredibly precise and dynamic process that ensures not only the equal division of the nucleus, but also the proper formation of a new, shared wall that integrates the daughter cells correctly into the plant tissue. #tag the movement of substances into and out of cells.
Okay, so we have these incredibly complex dynamic cells built and dividing, but they're enclosed by membranes. And you emphasized earlier that membranes aren't just solid bags, right? They're active players in controlling what gets in and out of the cell. What's the key principle behind their function as gatekeepers?
Absolutely.
Yeah.
That selective control is fundamental. All living cells are separated from their external surroundings by the plasma membrane. And as we've seen, eukaryotic cells like plant cells are further compartmentalized internally by the membranes of their numerous organelles. These membranes are far from being passive walls. They are selectively permeable barriers.
So selectively permeable, meaning they choose what passes through.
Exactly. They allow certain substances to cross much more readily than others. Some small, nonpolar molecules might pass through relatively easily, while ions and larger polar molecules like sugars or amino acids often cannot cross freely. Membranes actively regulate the amount, the type, and often the direction of substances moving across them. The key insight here is that this selective permeability is an absolutely essential capability for life. It allows cells to maintain their own unique internal biochemical composition and structure, which is often very different from their external environment. This internal stability is crucial for sustaining the complex processes of metabolism. Without this precise control over what comes in and what goes out, few metabolic processes could occur at the necessary rates, and the cell couldn't maintain its integrity. They also enable cells to establish electrical potential differences across them, which are vital for many processes like nerve impulses or energy generation.
Maintaining difference is key. And water. We keep coming back to water. It's the most common substance moving around. So understanding its movement is foundational to understanding all this transport.
Yes, water is absolutely paramount. Of all the different kinds of molecules surrounding and contained within a living cell, water molecules are by far the most numerous. And most other vital molecules and ions that the cell needs or produces are dissolved in water, forming aqueous solutions. Therefore, understanding the principles that govern how water itself moves across membranes and within the plant is really foundational to understanding almost all aspects of cellular transport and plant physiology.
Okay, so how does water actually move then? Both within the plant over long distances and maybe in and out of individual cells. What are the basic physical processes driving it?
Water movement, whether we're talking about it in living organisms or just in a non-living physical world, is governed primarily by three basic physical processes: bulk flow, diffusion, and osmosis, which is really a special case of diffusion involving water across a membrane. Let's touch on the first two from this section.
Okay, bulk flow first.
Bulk flow, which is also sometimes called mass flow, refers to the overall movement of a fluid, in this case, water, often containing dissolved solutes from one place to another due to a difference in potential energy. We often talk about the potential energy of water specifically as water potential. Water, like anything else, always moves spontaneously from a region where its potential energy, its water potential, is higher to a region where its potential energy is lower.
Like water flowing downhill.
Exactly. Gravity is one factor that contributes to water. Potential water at a higher elevation has higher gravitational potential energy. But another major factor, especially important in plants, is pressure. If you squeeze a bulb of water, you increase its pressure, you give it higher potential energy, and the water will move out. In plants, this pressure-driven bulk flow is the predominant mechanism responsible for the long-distance transport of sap. That's the aqueous solution containing sugars produced during photosynthesis, plus minerals and hormones within the specialized food-conducting tissue called the phloem. Sap moves under pressure from the source, like leaves where sugar is made, to sinks, like roots, fruits, or growing points where sugar is used or stored.
So pressure pushes sap around the plant in bulk flow, and this idea of water potential helps predict where water will go.
Precisely. Plant physiologists find the concept of water potential incredibly useful because it provides a single quantitative measure that allows them to predict the direction in which water will move, regardless of what specific factors, like pressure, solutes, or gravity, are causing that movement. Water potential is typically measured in units of pressure, like pascals, pa, or more commonly for plants, megapascals, MPa. By convention, the water potential of pure water at standard atmospheric pressure and temperature is defined as 0 MPa.
Pure water is 0. What about water with stuff dissolved in it?
Adding any dissolved substances solutes to water always lowers its water potential. Why? Because the solute molecules effectively reduce the concentration of free water molecules that are able to move. So aqueous solutions always have a negative water potential compared to pure water. This means water will always tend to move spontaneously from a region of higher water potential, like pure water, or a more dilute solution closer to 0, towards a region of lower water potential, like a more concentrated solution, which will be more negative.
Okay, water moves from higher potential, less negative, to lower potential, more negative. What about the second process, diffusion? That sounds like things just spreading out, right? How does that work and what's its role in the cell?
Yes, exactly. Diffusion is the net movement of a substance, whether it's ions or molecules, from a region where it is more concentrated to a region where it is less concentrated, simply due to the random thermal motion of the individual particles.
Random motion causes spreading.
Yes. Imagine dropping a small crystal of purple dye into a beaker of still water. The dye molecules and the water molecules are all constantly moving randomly, bumping into each other. Initially, all the dye molecules are crowded together. Just by chance, more dye molecules will randomly move away from the crowded area into the surrounding water, then will move back into the crowded area. So there's a net movement of dye outwards, spreading into the water. Similarly, water molecules will show a net movement from the area where they are more concentrated, pure water, into the area where they are less concentrated, where the dye is. This net movement occurs down a concentration gradient from high concentration to low concentration.
Down the gradient.
Right. The steeper the concentration gradient, the bigger the difference in concentration over a distance, the more rapid the net rate of diffusion will be. Diffusion is also generally faster in gases than in liquids, and it happens faster at higher temperatures because the molecules are moving more energetically. Eventually, if left undisturbed, the random movement will cause the molecules to become evenly distributed throughout the available volume. At this point, we say the system has reached equilibrium. There's no longer any net direction of movement, although individual molecules are still constantly moving randomly back and forth.
Okay, random motion leads to even distribution. What's its role inside the cell?
Diffusion is the principal way by which many substances move within the cytosol of a cell or across membranes if they are permeable to that substance, but only over very short distances. For example, oxygen diffusing into a cell, or waste products like CO2 diffusing out. However, diffusion is a relatively slow process, especially over distances longer than a few micrometers. For larger cells, like many plant cells with their big vacuoles, relying solely on diffusion to move substances around the cytoplasm would be far too slow to support active metabolism.
Too slow for big cells.
Exactly. And that's why active cytoplasmic streaming, cyclosis, driven by the actin-myosin system we talked about, is so important in larger plant cells. It actively stirs the cytoplasm, speeding up the transport of organelles and dissolved substances over longer distances within the cell. much faster than diffusion alone could achieve. So diffusion is fundamental for short-range movement, but bulk flow, like sap movement and cytoplasmic streaming, are needed for more efficient long-range transport within the plant and within the cell, respectively.
Okay, as we wrap up this deep dive, it's really clear we've taken an incredible journey. We've gone from the fundamental CHNOPS elements that, form the very fabric of life, through the intricate molecular machinery of carbohydrates, lipids, proteins, and nucleic acids.
The building blocks and the workers.
Right. And then into the complex dynamic world within the plant cell itself. We've seen how cells are the fundamental units, whether simple prokaryotes or the highly organized eukaryotes, especially the plant cell with its unique wall, plastids, and that huge vacuole.
And how interconnected they are in plants.
Yes. And we explored the intricate dance of cell division, from the precise choreography of mitosis to the plant-specific way of forming the cell plate. And just now, the critical processes of how water and solutes move, keeping these living systems vibrant. You should hopefully now have a much clearer understanding of how plants are built and how they function right down at this foundational level.
For me, thinking back over it all, what really stands out is the incredible economy and adaptability of life demonstrated at this level. How just a few basic elements combined into only 4 major types of organic molecules can be used to build such incredibly diverse structures and perform such a vast array of functions.
It's mind-boggling versatility.
It really is. From the immense structural strength of cellulose made from simple sugars, to the catalytic power of enzymes folded from amino acid chains, and the elegant information storage of DNA using just 4 bases. We've also seen how the plant cell itself, with its unique features, functions as a highly integrated dynamic system, not just a static collection of isolated parts. Every component, every process seems intricately linked, often with surprising dual roles, all geared towards ensuring the plant's survival and success in its environment. These foundational concepts aren't just abstract biology. They are truly the underpinnings of how plants build themselves and sustain nearly all life on Earth.
Absolutely. What stands out to you, our listener? For me, echoing that, it's the sheer interconnectedness, but also the elegant, almost ingenious solutions plants have evolved over millions of years. Things like those tiny isoprene molecules emitted by leaves, potentially helping regulate heat stress, or the absolutely critical role of lignin in allowing plants to conquer land and stand tall against gravity. Every detail feels like a testament to evolution's power.
Well put.
We really hope this deep dive has not only informed you, but also sparked even more curiosity, because there's always more to learn. For instance, think about this. How might a really deep understanding of the intricate balance of water potential in plant cells, something we just touched on from Chapter 4, how might that knowledge contribute practically to designing more drought-resistant crops, especially in our changing climate?
That's a great question, linking the fundamental science to real-world challenges.
Exactly. There's always more to explore, and the more we understand these basics from sources like Raven Biology of Plants, the better equipped we are to appreciate the larger wonders of the natural world, and maybe even help solve some problems. So thank you for joining us on this deep dive into the biology of the plant cell. Keep digging, keep learning.
And remember, knowledge is most valuable when it's really understood and can be applied. From the entire last-minute lecture team, thanks for listening.